We have one more panel here, and then we get to have a reception at 5:15. But and just I'll repeat it. It's an odd thing where the reception is. It's across from the reception desk through a blue room. So just remember reception all the way through, and you will get to the reception. So the final panel for today, which was just teed up by Beth very well, is the implementation of new research and technology in invasive species management. And it's going to highlight new and emerging technologies more than any other panel. It might be more of a show and tell. I know Heath Packard, who's here with Island Conservation, will be discussing the use of gene drives to eradicate invasive rodents on islands. I don't know how many islands there are in Nevada, but we'll figure that out. And this topic does have the potential to be controversial, so just take some caution. There are opinions about gene drive technology on the grounds it might get out of control and have unintended consequences, but we're going to have a very much controlled discussion and look at this. And I think that if you look at, you know, some of you are from states such as I am that has an ocean and oceans programs, and we've done the science on ocean acidification, which is pretty dramatic. We had a scientific study done that then did early action steps, and one of the early action steps was gene work to strengthen the shells of shellfish, which to a layperson, once again, was one of those you can do that moments, and how will that work and how will people feel about that. So those are some of the things that we're going to get at here. And so the speaking order today, we have Nicole Cartwright, the Executive Director of the Tahoe Resource Conservation District. We have the shy and retiring Dennis Zabaglo from the Aquatic Resources Program Manager from the Tahoe Regional Planning Agency. And he's the one that told me that I cannot use the acronym, so I just fully spelled out the name correctly. Doug Johnson, the Executive Director of the California Invasive Plant Council. Heath Packard, the Director of Government and Public Relations for Island Conservation. And we intend to follow the same format, opening presentations, some discussion, and then we hope for some time for some questions for you, so be thinking of them as we move along. With that, we'll go to Nicole.
All right, great. Well, thank you. Again, my name is Nicole Cartwright, Executive Director of the Tahoe Resource Conservation District, and I'm definitely doing a show and tell today about a really fun project that the Tahoe RCD was pleased to implement. It was a pilot project using ultraviolet light to attempt to eradicate or kill aquatic invasive plants in Lake Tahoe. And I'll just first kick off, because one of my funders is in the room, by thanking the two funders. The Tahoe Fund provided some seed funding in order to get the permitting and kind of compliance portion before the project went on the ground. And then the California Tahoe Conservancy, we acquired some funds through Prop 1 to actually implement the project. Also without recognition is, or not here in the room, is Inventive Resources. John Paluccio was an inventor who brought this technology to us and encouraged rigorously for us to put this project on the ground. And so really without his enthusiasm and passion for aquatic invasive species control and UV light and Lake Tahoe combined, this project would have never happened. Also want to just thank Lakeside Marina Beach Association for allowing us to use the project in their marina, the Tahoe Regional Planning Agency, and Lahontan Water Quality Control Board for their support in the compliance portion and allowing this project to happen. So all that stuff out of the way. Kind of get into what the project entailed. So here in Lake Tahoe, we are not able to use any pesticides or herbicides in the lake. So for the last 10 years, we've been pretty successful at using bottom barriers, which are mats that allow oxygen to flow, but they prevent sunlight from going down to kill plants, and diver-assisted suction removal, which has been labor-intensive, but it has been very successful. We have now about 5 locations around the lake that we considered to be kind of localized eradication. But we were looking for new tools, and that's why this project was so attractive to us. It was to get another tool in our toolbox without being an herbicide or a chemical control. So John Paluccio came to us with this idea about UVC light. It's ultraviolet light in the C wavelength, which is a short wavelength of UV. The idea behind this was that UVC light has been used as a germicide. How many of you guys have used maybe one of those little wands in your water bottle as you're backpacking to kill any bacteria that's in water as you're scooping it out of a lake from a high alpine lake? It's the same exact technology. So it kills one-cell structures. So the project that we did was here in Lake Tahoe at Lakeside Marina. There were a lot of assumptions and constraints throughout this project, but really what we wanted to know is, does UVC kill plants? There was a lot of laboratory work that John invested in before getting this field testing done, but some of the assumptions is that we're moving and maneuvering around boats. There's changes in temperature. There's changes in visitation. The area had previously been treated with both diver-assisted suction removal and bottom barriers, and so there was already previous investment in control efforts in this spot. So we have to take that all into consideration. And the other thing that we really needed to take into consideration is what other effects? So the whole point is to get this to be kind of in a programmatic method along with our other methods that we're using, but what other effects may UVC light be having on our water body, on the macroinvertebrates, on temperature, on DO, all of these other things. So that was the biggest part of this project, was not only just the treatment, but the monitoring plan that surrounded the project. So John and his craziness, who I love, he created this contraption that's basically just a pontoon boat that has a UVC lights on suspension that can lower down, and they sit on top of the plants. It's hooked up with all sorts of cameras and fish deterrents and all sorts of other widgets and gadgets, but ultimately it's just a pontoon boat with a giant light. That's what it comes down to. Here's again some of the original drawings of what the vessel was going to look like prior to being built. Glad I printed this because I can't see the screen at all. So in the water, this is what it looks like. A lot of adaptations happened throughout the process. When we got the boat up here, we realized that pollen was a huge issue and that pollen was capturing onto the actual UVC lights underwater, and so time intensive to go in there and clean off the algae, clean off the pollen every day, day in and day out. This version is actually the later version that he was then folding up at night, so it had a longer extension arms on either side that were folding up, and then once it was treated, those levers were sort of let down, and so it kind of reduced those issues. And just to get a quick glimpse of what this looks like, the picture on the left is been treated, on the right has been untreated. That's with one pass of UVC light, varying lengths of time, 5 to 20 minutes per section. And if I should have gone back actually really quick. So, sorry, those boxes are very, very tiny, but those little red boxes are his treatment plot areas, and everything was tracked with how much time each site had, what plants were where. So I should have mentioned that. All right, so visually it works, right? But now we have to do all of the follow-up. Here's just another version of this. This is actually in the open water, or what we call Lakeside Beach. Everything within the white PVC was treated with UV light. Everything outside of the white PVC was treated with diver-assisted suction removal. The bottom pictures showing 2 months later, both sections that have now been treated look identical, right? So plants are gone in both locations. some more visuals. I'm going to use the word very loosely because there has been no paper yet, but the idea of collapsing or decomposing or dying. But you're seeing here basically before treatment, Eurasian water milfoil and curly leaf pondweed, which are our two hot species here, and then progressions 2 weeks after treatment, 4 weeks after treatment, and 6 weeks after treatment. Our protocols in this project specifically required us to have monitoring during treatment, and then we also had this season in 2018 post-treatment requirements. So we had 1 full season of post-treatment requirements this year with no treatment at all in the second year. Again, I'm using the word collapse very loosely, don't quote, but here is just a graph of kind of the time it took for each plant species to collapse or die back. The two, I don't know if the colors are coming out, but the one that dropped the quickest was leafy pondweed. It's a very kind of weak or wafy plant structure. And then next was Eurasian water milfoil and coontail, a little bit more robust structure. The one that collapsed last was our Richardson's pondweed. It's actually a native. It has a very robust leaf structure. So likely, it's harder for it to break down from the UVC light. In approximately 10 to 15 days for all the plants to collapse. So what's next, I guess, or what did our monitoring look at? There is a progress report that is posted on our website, tahoercd.org, that had all the findings, just facts, no assumptions were really created there. From 2017, that's on our website now. We are working with our consultant, Mel Green, actually in the room as well, to answer the rest of the questions that are unbolded. So that's kind of the next step, is to answer all of these questions. There will be the final report coming out in December or January, depending on the review period. So just some initial results, and lessons learned. Treating the plants earlier would have been better. By the time we had funding in hands and permits in hands, and all of the agencies in compliance, it was pretty late in June when we got the treatment started. So it would have been easier because plants would have had less density. They wouldn't have been so tall to treat probably in end of May or early June, so just a couple of weeks before. The vessel that was created for this and built for this was just a pilot vessel. So there's the possibility of ramping up, building a larger vessel, being able to treat more area at a longer period or for more time. With a larger vessel, we anticipate treating approximately 1 acre per day in open water. So that's not getting congested with all of the different boats and marinas and piers. There's a lot of factors into what constitutes really good treatment or timing, and that really the lesson learned is that short wavelength light, UVC light, works as a method to kill single cell organisms. Our next steps right now are, and I know we don't have Q&A, so don't actually ask me any questions, but our next steps right now that we were thinking ahead, and that is the one great part about our partnership, just to highlight that what they were talking about this morning, is that we have already received funding, and we are working on changing our programmatic environmental documentation to include other mechanisms. So right now we're approved at a programmatic level for bottom barriers and diver assisted suction removal. We're moving that document into a larger document to then include UVC light and some other technologies. So we're kind of, even though this project isn't complete and we don't have our findings yet, we're moving into that next level to hopefully put this on the ground in about a year or two as a programmatic level. So thank you.
So thank you very much. Dennis?
Yeah, thank you. Thank you, Secretary. Thank you, Nicole. Dennis Zabaglo with the Tahoe Regional Planning Agency, not TERPA, Mr. Secretary. Thank you. We've evolved from that. But I wanted to kind of build on Nicole's theme of the tools, having the tools in the toolbox, and how all this fits in to our larger control strategy, dealing with multiple invasive species that we currently have, but also leading into prevention as well. So you heard Governor Sandoval this morning mention the Tahoe Keys. It's a homeowner area on the south shore of Lake Tahoe. It was once the largest wetland in the entire Sierras. It's about 172 acres of channels and waterways, 1,500 homes, all infested with Eurasian water milfoil and curlew pondweed. So it's representing a much, much larger problem that we've tackled and had success with to date. Some of the other projects that we've had success with, that Nicole mentioned, with some other methods, non-chemical methods, bottom barriers, hand pulling, that's been really great at a small scale, 2, 3, 4 acres per project. But now when you're talking about 172 contiguous acres, we're bringing a knife to a gunfight, so to speak. So what else can we put in that toolbox or that armory to give us greater success? And so that UV light is certainly one of the things we're looking at when we're talking about trying to have all those tools in the toolbox. So you see here, this is the Tahoe Keys. It represents some of their current activities, basically from a maintenance standpoint, mowing, and that it actually makes the problem worse, but it's basically done for recreational purposes. The mowing just cuts all the weeds down to a boatable level, but all those little fragments that are created make the problem worse. All those little fragments can turn into a new plant. And so what else can we do to help control that? And the picture on the left is representing the hot spots where curlew pondweed now is kind of taking over Eurasian water milfoil even. And that's scarier for us because it has a greater propensity to live outside of these kind of entrained areas out in the lake proper, can resist wave action. The way they reproduce are with these little turions that can sit in the substrate for much longer and may be resistant to some of the bottom barrier methods that we're talking about. So in addition to UV light that Nicole mentioned, we're looking at other techniques for different invasives, but also one that's happening right now within the ski run marina, and it's a completely privately funded project using laminar flow aeration. It's essentially just taking lots of little bubbles at the bottom in the substrate with these ceramic diffusers and adding oxygen to the entire system from in the substrate all the way to the surface, creating an oxygen-rich environment and basically trying to stimulate that the muck layer at the bottom to decompose. That's the food for plants, and so if we can have an effect on reducing the food, then maybe plants will be reduced. We've seen this in some other places in the Midwest where that's been a byproduct of the technique, where that's usually used to just add to oxygenate water bodies. But now is this another tool we can utilize, and so it's being tested right now. So hopefully we'll have some good information on that next year. Also with Asian clams, the governor mentioned that today as well. It's a pretty big infestation for us from the south shore all the way up past Glenbrook on the east shore of Lake Tahoe, using bottom barriers again to suffocate them. But for us in Tahoe, aesthetics is certainly a big deal. It's one of the most beautiful places on the planet, and we want to try to protect that. And so the techniques we currently use leave the dead shells behind. And so, like I said, it's an aesthetic issue, but could they also be creating a more calcium-rich environment in the water column that would be more conducive for quagga mussel infestation? They need a little bit more calcium than Asian clams do, and so when these shells are decomposing, do they add that calcium that makes it more suitable? So one thing that's being looked at, that picture on the bottom, is a clam harvester. It's a company out of Canada called Aqua Treasures, and they are developing a technique that basically sits on the bottom and moves across the bottom, separating the clams from the substrate and returning the substrate back down. So if you're a hockey fan, clamboni has been a term utilized rather than a zamboni. I think it's pretty clever. I'm a hockey fan, so.
I think Western Governors is going to be doing a glossary after this, including things from the different panels.
But I have to give credit to Zach Hymanson, who's with the Resource Agency now. He came up with that term, but I thought it was super clever. So that's being tested, I believe, this month or next month up in waters closer to where they're from. It's a little bit hard to mobilize, and we're hoping that then we can utilize that, test that at a larger scale down here. So that's how a lot of these things we learn from, right? We do small pilot scales and then upscale them. to larger projects, and that's certainly true with Asian clam. And then I also wanted to mention, oh, kind of duplicated there, but looking at innovative ways on the prevention side as well. The picture on the left is from a Volvo outdrive. So it's the portion of the engine that sits on the back of the boat. The other kind of car engine part is on the inside. But the boat industry has been really stepping up their game with trying to deal with invasives. We all have the same customers. Customer service is a huge aspect. We want people to recreate. And when they come to do boat inspections, it's time, it's money. So the boat industry has recognized this as well. If it's becoming more complicated, more costly for boaters to boat, maybe they don't buy boats anymore. And so the boat industry is taking steps to help us. So we're sitting at the table together. They're very active and engaging. We've had great collaboration. So this is something Volvo's done. If you can see the little blue thing on the bottom left there, pretty low-tech type of thing, but it is technologically advanced from what used to happen. When we do decontaminations, it's these little earmuffs that go on the outside of that drive there, and we're just flushing the engine with hot water. It's not a perfect way to do it. Sometimes it's not all that efficient. It works, but sometimes it's challenging. And so what Volvo has done is just add this little quick connect. It's like your air compressor or something like that, the little quick connect that purely facilitates decontamination. So they've done that to their boats just for the purposes of aquatic invasive species. So that's really encouraging. I'm actually leaving tomorrow afternoon, right after this, to go back east to work with the American Boat and Yacht Council and National Marine Manufacturers Association to kind of continue this progress. We've developed what's called a technical information report for the boat industry. It's an education piece that the resource managers and the industry wrote together to educate them what we're doing to their boats, putting hot water. And when we first had what we called the AIS Summit about 4 years ago now, their first reaction was, "Oh, you're putting hot water where?" And we're like, "Well, it's okay. We're not using chemicals or anything like that." And so getting that information across to them, educating them why we need to do this, why it's benefiting their boaters to have these inspection programs in place. And so this information report educates the boat industry on what we're doing, why it's a problem, and what could they possibly do, like this example with Volvo, to design and construct boats a little bit differently that makes it better for all of us in the long run. So, thank you.
Thank you very much. Doug?
Thanks, Dennis. Thanks, Secretary Laird. So I was a mechanical engineer in a former life, and I'm digging all these cool technological solutions for killing weeds. I'm going to talk about innovations that are partly technical, but partly not. And I also want to remind us that there are innovations in communications, et cetera. Like we talked about earlier, I had something come across the email this week that was on environmental humanities, and it mentioned the problematic construct of invasive species. So I'm guessing most of us have to deal with that with some audiences, and I think we should be looking for innovations kind of outside the technical box too. People have controlled weeds forever, so like what's really new about it? And of course there are new and better ways to kill weeds. Herbicides are developed that are more selective. Biological control agents are tested for host specificity in more thorough ways. So some of the tools are getting better, but what I want to talk about is actually not rocket science, but it's taking the concepts of strategy that we all know, kind of the basics of getting things early, getting outliers, pioneer populations, and how technology is enabling us to do that better and to do that at a larger landscape level scale, and to incorporate new data into that about everything from climate change to wildlife corridors, things like that. So, for instance, we have a brand new weed in San Diego County and Orange County in Southern California, desert knapweed, and we are able to look at that and say, how far do we think this could spread? And that's possible because we have climate data from around the world, and can apply that to extrapolate from where something grows now to where it might grow. So in this case, seeing that this could grow widely across the Southwest, that's a pretty compelling case for a rapid response to this early detection. Now this is a close-up of that same major infestation in Borrego Springs, down near Anza-Borrego Desert State Park. And you've all probably seen data sets online where people have mapped things with their smartphones, for instance, and uploaded points to an online GIS mapping platform. And so this is CalFlora, something we use in California. It's similar to EdMaps and IMAP invasives and other systems that are used across the country. And what's important about this, of course, you need to know where the weeds are to control them, but this is from hundreds of different volunteers and different agencies, all compiled into one map. And that's critical. That is some of the work that WGA is also working on to help us aggregate between those systems like EdMaps and CalFlora across state lines, so we can see at the landscape scale where things are and where the opportunities are for controlling them. This is a screenshot from Calypsy's online decision support tool called Cal Weed Mapper, and this was developed with support from the Forest Service and from the California Landscape Conservation Cooperative. And this is both a technical tool that can show you where any particular weed is and isn't. The isn't is probably more important than the is in California. And it can also show you things like what's the suitable range for this plant today, and how does that suitable range change in the next 50 years. And that's extremely useful information when you're trying to set a priority at the landscape scale. One of the things this tool is designed to do is to generate a management plan or a draft management plan. Humans still need to look at it and decide what makes sense, but to tell you for a given user-selected area, and I think on here a county is selected, San Benito County, if I'm not mistaken, and tell you which weeds are just getting started there and are maybe eradication targets, which weeds are widespread and may be control targets, and which weeds aren't there but are within a certain distance, like 50 miles, and maybe EDRR, early detection targets. So the next steps with this are to add some of the brand new data sets that are being generated from California State Wildlife Action Plan. We've got the vegetation macro groups and how those tie into sensitive and listed species. And so we can begin to look for interactions between the invasive plants and the impacts they have on the ground. As Dr. Ledger said earlier, we've got a couple hundred recognized invasive plants in California, and limited resources to understate the situation to go after it. So where do you work and on which plants? And so these kinds of data sets will really help us land managers figure out where to work. So one of the other things, whoops, I think I went to, so one of the other mapping, sorts of mapping data we have are aerial imagery that has just improved and improved and improved to the point where it's scary, and you can look at your own house online and see yourself standing in the backyard or something. But the good news is you can see large weeds, and so one of the major weeds we have in California, and it's a big problem in other states as well, is a rhododendron or giant reed. It's a 20-foot tall grass that grows in riparian areas, and to map that across the state on foot would take a lot of time, but with aerial imagery, we're currently able to, with a Prop 1 stream flow enhancement grant, to map this plant across 14 million acres of the Central Valley. And that sets up potential control projects, so a local RCD can take on a whole watershed and know that, okay, I want to get the upstream parts first. Here are where all the populations are. Here's how much total acreage we have. Here's whose land it's on. So that aerial imagery is a very important tool as well. And then we know that this is a dynamic problem, invasive species. We've got all the ones we've got now. We've probably got some we don't know about yet, and we've got more coming in the future. So these are 4 plants that have been assessed using a risk assessment. It's probably too small to see, but this is the plant risk evaluator developed by UC Davis and the University of Washington, primarily developed initially for horticultural screening. So if you're bringing in a new plant, or you want to bring in a new plant, take a look and see if you can predict how risky that plant may be for being invasive in the future. So you're looking at its life history, its biological characteristics, and how it's behaved elsewhere in the world. And you get a score, a numerical score, and that puts it into a category of either, hey, should be safe, or no, don't do this, and then there's an in-between gray area too. We have used that to predict which things that have already escaped into California's wildlands but aren't spreading yet, but land managers have seen them, which of those things are the highest risk to become a problem in the future. And so we also, with funding from the Forest Service, looked at some 200 plant species and came up with 80 of them that are a high risk for becoming invasive in the future. So we've now added that to the CalIPSI inventory, which to date has only listed those plants that are known to be causing problems in California's wildlands. And we've now added these watch plants, which are not invasive, but they are ones to really keep your eye on, because if you start to see something happening, we want to get on them early. So I believe that is the last slide. I do want to say that in addition to all of the innovations happening out there, of course, it's important to get the information out to people, the tech transfer. So I know a lot of your organizations do this as well. We have an annual conference, we have a newsletter, and I think, as John Kabushima mentioned earlier, the new Invasive Species Council, or the newly codified Invasive Species Council in California, provides a communications opportunity to really make sure that the public is even more aware than they are now about the problem and some of the innovations and ways that we can go after them. Thanks.
Thank you very much. Heath?
Good afternoon. I'm the sweeper, the cleanup. I appreciate this opportunity.
What's the sweeper do in an invasive species form?
I appreciate the opportunity. Looking forward to a good dialogue here as we close out the day and on into the reception. I'll try to be quick so we can have some dialogue. Island Conservation, we are the world's only global conservation organization whose sole mission is to prevent extinctions by removing invasive species from islands. Why do we focus on islands? Because that's where most extinctions have occurred. That's where most endangered species are, and invasive species have been implicated in most of those extinctions, and they are causing serious risks to the birds that we care about, in addition to the economies that we heard about, the economic impacts, human impacts, health risks, etc. So we focus where the confluence of extinctions and the opportunity to have the greatest impact. Why are we here? We're here because we were started by a couple of postdocs at UCSC, Santa Cruz, 25 years ago. We've been partnering with the U.S. federal government, many partners in this room, states, and local communities for years. We brought new technologies and have innovated technologies from New Zealand, where eradications of invasive species on islands began, and we've applied them here. And we're working every day to try to do it better, faster, quicker, safer. This is a picture of Midway National Wildlife Refuge, which we hope may be one of our next projects. The U.S. Fish and Wildlife Service is going through an environmental assessment right now. There are literally millions of albatross on this island, and it's infested with mice. And here's a little disclaimer, just for those weak of heart, you might want to avert your eyes. The rats have been eradicated from this island, but there's mice on the island, and they've learned to become carnivorous. And the albatross are so committed to rearing their eggs, incubating their eggs and rearing their chicks, that they will not leave the nest when the mice climb on their backs and start eating them alive. And so we're losing lots of albatross very quickly. I'm going to move quickly beyond that. So how do we do eradications? This is a message of hope. We're very excited because there have been thousands of successful eradications of invasive species from islands, and lots of new technologies and tools that have evolved that are giving us new tools every day that can have implications for the mainlands, for our interior states, as well as our coastal states and our islands. But we've got challenges. To get rid of rodents, we have to use rodenticides. That's the only tool that really works. And this has been done 500 times around the world, and successfully. But it takes time because the rodenticides are not discriminate. They're indiscriminate, so you have to figure out what the risks are to the food web, how to mitigate those. In Midway, we're going to have to collect the lace-on ducks that are there and make sure that they're safe while the rodenticide's on the ground so that we get every last mouse, but we protect the ducks that are there. And these things take time, they take money, etc. Given that there's rodents on 80% of the world's islands, and there's more than 400,000 islands across the globe, we've asked ourselves, what can we do to really increase the scale, scope, and pace of eradications and advance the movement of controlling, ultimately eradicating invasive species? Islands are a great laboratory for this. Some people will make the analogy that an island, or lake, if you will, or a watershed is just an upside-down island. When you have a geographically contained space, you might be able to do some pretty interesting things in there as these tools and technologies come on board. So, we're looking for transformative innovations. We're also looking for incremental. So in terms of the bait application, which is done aerially on big islands, we're actually working with a company in Germany to look at drones and doing smaller scale, more precise application of this bait aerially so that we can reduce the risks, increase the precision, increase the effectiveness, reduce the cost. But transformative innovations, we're trying to find some areas there. And why are we doing that? New Zealand has declared that it's going to be predator-free, rats, mice, and stoats gone by 2050. They don't know how they're going to do it. The main islands in New Zealand, huge islands, tons of people living there, built environments, cities. If New Zealand can do that by 2050, maybe we can do that in Hawaii. Maybe we can do that in Guam. Maybe we can start looking at eradications on the mainland. So this is really exciting, cutting-edge stuff that we're talking about. But it's going to take time, and it's going to take some vision, and it's going to take some exciting new ways to get about this. In the U.S., we've been working with the federal family for years. We have a new partnership that we're developing. We're calling it the U.S. Island Partnership with a big 2050 vision. What if we could have all U.S. federally owned and managed islands invasive vertebrate free with biosecurity plans in place and restoration plans in place by 2050? Can we do it? We don't know, but if you set a goalpost and you start working towards it, you start thinking big. That's led us to doing a horizon scan, which was published a few years ago. I can show you the paper. We looked at all the emerging technologies that might help us with doing eradications on islands, and all sorts of interesting things going on. And one of the areas that we got hopeful about, but had a lot of questions about, was the possibility of gene drives. This slide shows you, brings you back to 10th grade genetics lesson. Pink ears versus blue ears, Mendelian genetics. Typically, genes are distributed about 50-50 through natural selection and sexual reproduction. But a gene drives are found in nature, and they come from nature. The mouse actually has a native gene drive in it, a natural gene drive in it called T-complex. And there are gene drives that come from other parts of nature that geneticists are using to insert into different species to also see if they can bias the selection of a particular trait. So we have researchers that are looking at this and trying to figure out, could we do this? Could we create a self-limiting mouse that would drive through sexual production, drive male sex selection through multiple generations so that after a year, year and a half, 2 years of sexual reproduction, after you've introduced this mouse to the island, to the invasive mouse population, you would have one population that was all male. They'd live out their merry days. There would be no lethal control. There would be no need for rodenticides. This has piqued our interest. As such, we've formed a partnership called the Genetic Biocontrol of Invasive Rodent Partnership. 7 institutions from 3 continents, and we are exploring as thoroughly, comprehensively, and holistically as we can whether this is possible. We're asking ourselves not only could we do this, but should we? And if we could, and should we, but under what conditions? So we always work with local communities, their governments, to figure out if and when and how our values line up to do an eradication. This would be the same case. But in this case, we know we have a lot of unanswered questions that need to be addressed. We need to make sure that this is able to be done safely, that it's able to be localized, that we have safety mechanisms that can make sure that if there is escapement, we don't have unintended consequences. We know that social acceptance is going to be a real challenge and something that we need to work with communities at the forefront. We may be 10 years away from this technology actually being feasible, but we have, as you can see, all these various boxes, about 15 different investigations going on to figure out if this could be done, should it be done, and under what conditions. There are other folks working in this space. This is a picture of honey creepers that have gone extinct on the Hawaiian Islands. And today, they're facing more extinction. More of the forest birds are facing extinction from avian malaria. Folks are working in mosquitoes to try to target malaria, human malaria. Could this be done for avian malaria? People are working on chestnuts. They're working on ohia trees. Could the American chestnut be brought back with genomics? So these are some interesting ideas that we need to pay attention to. We need to dialogue as a community. We've seen some biocontrols work. We've seen some biocontrols go very, very wrong. We need to learn from that. We need to evaluate, could this be done? If so, under what conditions, and how would we do it? This is just a schematic of how the mosquito might work for a self-limiting mosquito. I digressed there for a minute. So with that, I know there's lots of questions. Look forward to talking to you about them. And thank you very much for the opportunity.
Thank you. A great introduction. And we just have 20 minutes left, and I want to get to questions. But first, let me just ask a question of the panel about how receptive you find that maybe invasive species professionals are for some of the different things that you've talked about. Open arms or a real fight to get them interested?
I think it's a bit of a mixed bag. I think there are some that don't fear change, but others that do. I was at a conference recently where the UV light technology was presented, and there was quite a bit of criticism regarding it, but a lot of optimism at the same time, so it's a mixed bag.
I'll just add on that. We've definitely heard the same thing as we were piloting the UV light project, that there was some skepticism, and someone in an earlier panel said we all just kind of get used to what we know and what works well. And so stepping outside that box into something new, and I think also with the industry, herbicides and the amount of money that is in that industry, I think it's scary to some that there could be alternatives. So yeah, I think with anything, it takes time and it takes change and it takes good science to get behind it to make sure that folks feel comfortable. So I appreciate what Island Conservation is doing with all the background.
And on top of that, we have limited budgets, and so when we have to affect change with a small dollar amount, you kind of tend to go to what works without taking that risk or leap of faith to investigate.
Well, that's one of the other questions I had to ask. Oh, no, it's perfect. You just teed it right up. Which is, some of these technologies are just very expensive to implement. How do you balance that, or how do you make it work? Were you about to add something else?
I was just going to say, yeah, in addition to learning to do something new, if you're doing anything like planting instead of just killing weeds when those resources are so limited, there's also pushback sometimes for that, rather than try to learn something new or to plan a strategy that might be more effective. Spending money on that instead of just going out there and putting some money on the ground can be counterintuitive too.
Well, go ahead, Heath.
I think we know we need to pursue efficiencies as well to try to save money and save time. So on the biosecurity front, one of the innovations we're looking at is machine learning, to have computers be able to analyze the camera traps that we set up for biosecurity purposes can we get that early detection done faster? Can we set ourselves up to be able to respond quickly for a rat spill on an island so that we don't have to go through really long and time-consuming permitting processes, but we can get the incursion when it's going to cost the least amount and have the least amount of impact. So those are other ways to save time.
You know, and let me just ask a follow-up. One of the things, you know, I spent 8 years working on dams overtopping and a drought and fires and really busy, and I had this small bucket list of things to do before I left. And I went out to the Channel Islands last year, and they had eradicated, I can't remember, rats from one complete island. And then there's some question about whether the current administration might backtrack on some of the things. Is that an issue that sometimes you might have some success and people might want to not appreciate it as a success?
Certainly a challenge to sustain healthy and restored island once you've done an eradication. Reintroductions are a challenge, so that's why you need good biosecurity in place, and land management decisions as they move forward can, you know, have different implications. And so working in partnership with the land managers and the local communities on shared values is probably the best way to sustain good management moving forward.
Then let me ask, in a moment we'll go to questions, but did anybody have anything they wanted to add on the money issue and whether some of this is too expensive to design and implement, or what you have to do to get past that? I know Heath spoke to it. Is there anybody that wants to add anything?
I'll just say it's definitely being addressed in our pilot project to not only look at is the method effective, but is it also cost effective compared to the current methods that we're using. So that will be a part of the analysis to look at that.
And you need to look over the long term too. I think it was Paul who said earlier, you know, frankly, it's not necessarily going to, you can't necessarily sell the concept of, well, this is going to reduce fire. You'll see it next year if you just put some money into this. It's a long-term maintenance problem. So it needs the kind of funding that, I mean, you have to say, yeah, we're going to bite off investing some up front because some of these ideas are going to result in more efficiencies and better conservation down the line.
Right, good point. It's an opportunity for people to ask questions. Who has a question of anyone on our panel?
What's at the happy hour?
You're between us and the bar. It's always the worst place to be. I just love everything that you guys are doing. This is super exciting. And so we worked really closely with Nicole and Dennis, obviously, on the UV light and some other things. The question that sometimes we get is, and especially when you're in the innovative space, is, and I know Beth talked about this earlier, sometimes you have to kill off a little bit of the native in order to really take care of the non-native. And do you guys have thoughts on where you stand on that, and what's an okay percentage, or how do you manage that? Because to me, that's really the challenge when you're introducing something new, is that there could be a side effect, but maybe the side effect is kind of worth it if you're going to tackle something bigger.
Yeah, I think that, yeah. We have lots of regulations, especially in California. We have a CEQA process that we go through that identifies some of those things, but ultimately the question is, what's the cost of doing nothing? If we're not having, I think, bycatch or something like that, depending on what the project is, if we are killing a few desired species in some of these trials, if we're not doing anything, they're going to die anyway by being taken over by that invasive species. So I mentioned earlier, we kind of take these small scale approaches, find out what that undesired byproduct is of killing a native species, and how big is it? Are there other things we can do to control it? What is the response of the native once the technique is employed? And to a lot of the treatments we're doing here in Tahoe with the bottom barrier specifically, and Nicole, I think, could probably speak to this more, but we're seeing that the native populations rebound pretty aggressively after an invasive is treated. Even though there may be some pockets of the native plant within those communities, they're bouncing back. Or kind of habitat restoration, I guess.
Yeah, we have a similar experience. We pursue projects with partnerships in the communities and their governments, and in the U.S. we do have pretty rigorous regulations that help us ensure that we're avoiding, minimizing those risks. And we only pursue projects where collectively we agree that the potential for short-term risk is significantly outweighed by the long-term benefits. And that's proven out in almost every project we've ever done. And we've got remarkable recovery stories to highlight.
That's perfect, because one of the questions I was going to ask is risk versus rewards. You answered it without me having to ask it. Other questions? Who else has a question? There's one right here.
I'll ask the gene drive question. So the big concern really, of course, with gene drive is that you're able to push that gene through not just a localized population, but if it escapes, say, the island and gets to another island or another location, or even back to the native, homeland for that species. So how do you control and prevent that from happening?
Mm-hmm. That's exactly one of the key questions that we're endeavoring to answer. There's research being done. So there's disputes in the science right now as to how strong these drives will actually be. We have researchers who are concerned that they may not be strong enough to actually work to do what we want them to do on the island because there is natural selection against them. But despite that, we're going through efforts to figure out how to do it safely by linking it to local alleles. So there are genetic unique aspects to an invasive population on an island that are going to be unique to that island only. So if you hitchhike your gene drive to that, and that mouse construct were to get off the island, it wouldn't actually work with a mouse that doesn't have that same local allele. There's other approaches where they're doing daisy chains, where you have to have a combination of 3 different elements come together. There's other projects looking at, are there shutoffs? Can you introduce an alternative that would just shut it off? So those investigations are going to run their course until we have high degree of confidence, virtual 100% confidence that we can safely control these things. The nice thing about islands is that we do have a biocontained environment that we can do a fair amount of biosecurity around, and if something goes awry, we could revert to old methods if we need to. So there's a number of safeguards that are being built into our thinking if and when we get to the point where we ask ourselves, do we know enough, are we confident enough to try a field trial?
Great. Another question. Does somebody else have a question? Then maybe getting to a close, let me just ask a combination of 2 things I was going to do and just see if you think there are any technologies we haven't talked about already that are on the horizon that might really prove the ability of this, and if any of you wish to really go for it, is there a moonshot here somewhere? Is there something that would just be so transformative that we have to get there?
I am not an expert on eDNA. Maybe some of the aquatics folks here could speak to that, but that seems like something that's allowing early detection in a way that hasn't been possible before.
I'm not going to be able to speak very intelligently on this at all, but it was a pretty impressive research that was going on. I think it started more for cancer treatment, but it was researchers at Stephen F. Austin University. They were creating an endocide, so doing something with the endocrine system to kill itself, so a cancer for a cancer, so to speak, where they were trying to develop something with Eurasian water milfoil that it was just the essence of the plant itself that they could introduce into a system that only impacted that species. Not necessarily an herbicide, but using, almost like a vaccine, I guess, so to speak, the best way I could describe it. Like I said, I can't speak very intelligently on it, but if somebody can look it up from Stephen F. Austin University, is something I'd really like to learn more about because it seemed very exciting.
Great. Heath or Nicole?
I think what's most exciting is that this community is starting to ask those kinds of questions, or asking them more often, and really digging in. doing those horizon scans. There's RNA interference that folks are investigating. There are radius-specific toxicants that work. They just aren't palatable. So if somebody could figure out how to make that tasty to a rat, that could be a breakthrough. And we mentioned some of the machine learning, artificial intelligence to help identify early detections. Drones the size of these round tables that can carry a payload of bait, and genomics are some really interesting areas that this community should and can and should be investigating, leaning in on, and looking to do other horizon scans to see what's next, because these are not intractable. It's just a matter of time before we find out what the solutions are to get to them.
In answer to your question, I would say I don't know of a moonshot, and I would hesitate to think, in the mindset that technology is going to fix this in some way. I think it needs a massive effort, lots of people, lots of money.
I also would, this idea has been brought up before, but something that policymakers could help with is bringing together inventors, because we are in the trenches day in and day out doing natural resource management. This gentleman that brought this idea to us about UV light had the idea because he was trying to treat algae in caves, and this is a guy that came from the stormwater world making infiltration systems for drains. I mean, these are the type of brains that we need to bring to the table and have those discussions outside of, the scientists and the biologists and the wildlife people.
And I think the Great Lakes just finished an award program where they had, I don't recall the dollar amount, $100,000, $500,000 for people to come up with new ideas for quagga mussels. And so having like a shark tank, so to speak, might be a way to do it.
We've worked with the National Invasive Species Council and Conservation X Labs on a couple similar approaches. And so creating those digital makerspace opportunities where you can get creative, innovative young folks who are willing to take on a challenge for not a ton of money or prize money is a great approach. And also thinking about financial innovations, like how can we fund this? How can we find new and creative ways, looking at the finance expertise that we could tap to find ways to invest in these things in the long run, because it's going to save taxpayers money on the back end.
Well, great. This has been a great panel, covered a lot of ground. Join me in thanking Nicole, Dennis, Doug, and Heath. And just the two quick housekeeping things.